Hello everyone, welcome to CUET PG lesson session. And in this session, we are going to see the previous year question paper of 2022. Okay, so let's move on to the session. The first question, which of the following technique is used for tracing a change in intercellular location of proteins and metabolites? So the question is to identify the technique which is using for the tracing the protein location and the metabolites. So they have given four options: chromatography, DNA footprinting, DNA end labeling, and pulse chase experiment. So the chromatography is for the separation technique. Okay, for the biomolecule separation, this chromatography is going to be useful. And the DNA footprinting is for tracing the suspects for identifying. It's completely based on the DNA. Okay, and the option C, DNA end labeling, depends upon the DNA. The final option is pulse chase experiment. So only this is going to use for the tracing of location of protein and the metabolites. Okay, so the option D will be the correct answer. Okay. Okay. And the next question, a specific protein required for infecting host by coronavirus is known as. Okay, so there are four options: receptor protein, PDE, spike protein, and the polymerase basic 2. So usually the virus will have the outer protein which will be helpful to attach on the host. Okay, so those kind of proteins are always a spike so that it can be easily attached and where the DNA can easily it can be penetrate into the host. Okay, so the correct option will be spike protein. Okay, option C is the correct one. And the next one is the most common initial step to separate water-soluble protein from insoluble cellular material. So they have given four options: electroporation, differential centrifugation, density gradient centrifugation, and the Western blotting. So let's go on to the first option. So what is electroporation? It is a separation technique by giving a electrical field. Okay, so this option won't come. And the next, we will move on to the third one. So what is density gradient separation? So it is going to be separate based on the weight of the particles. Okay, so but the question they have asked that water soluble proteins from insoluble material. So we are not separating two different things. It is going to be separated based on the density, weight. So the option 3 was also not correct one. And the last one, Western blotting. So which is useful for identifying the specific protein. Okay, so the last one is what? The differential centrifugation. Option B will be the correct answer, which is a first step that is used to separate the soluble protein from the insoluble one. So once we have separated the molecules, after that we will go for the electroporation or for the Western blotting techniques. Okay, so for this question, the differential centrifugation is the correct answer. Okay, and the next one, the pH at which the net charge of protein becomes 0 under the electrical field at room temperature is known as. Okay, so when we are placing a protein at under the electrical field at room temperature, it becomes 0. Why it has become A? Because due to the isoelectrical part. Okay, so protein has the nature of switching on to acidic nature as well as the basic nature. Whatever switching happens, it will be the charge will be remains 0. So the option will be D, the correct option, D, isoelectric point. Okay. And the next question, the microscope that converts a slight difference in the refractive index and the cell density into easily absorbed difference in the light intensity is known as. So there are four options: phase contrast microscope, bright field microscope, dark field microscope, and the transmission electron microscope. So the transmission electron microscope, they will use a electron beams as a light source. Okay, so the option D won't come. And for bright field and dark field, the light source will be narrow. Okay, so there won't any changes in the light intensity. So this two option won't come. The last one is phase contrast. Option A is the correct answer. So by changing the light intensity, we will get the different images of the specimen. Okay, so the microscope that converts a slight difference in the refractive index and the cell intensity to observe the difference in the light intensity. So we will observe the contrast images. So with the help of phase contrast microscope. So the option A. Okay. And the next one, which of the following histone protein found in chromatin has high lysine content? They have given four options: H1, H2A, H3, and H4. So the correct option will be H1 because H2A, H3 and H4 will playing an important role in the transcription or for stabilizing the chromatin content, for protecting the chromosome. This other kind of histone protein will be playing. So the option A, H1 is an correct option, which is having a high lysine content in the chromatin. Okay. And the next question, which of the following techniques may be used for identifying a regulatory protein that acts by binding to specific sequence of DNA? Okay, so in the question itself, they have given protein and the DNA. Okay, and identifying the technique. We need to identify the technique with the protein as well as a DNA. So, okay, so there are four different options: southern blotting, northern blotting, southwestern blotting, and western blotting. So southern blotting is for what? It's for a DNA identification. Okay, we are going to identify the specific sequence of DNA. And the northern blotting is for identifying RNA. Okay, and the western blotting, option D, is for completely for the protein molecule. So the remaining option is what? Option C, southwestern blotting. Okay, so we are going to identify the regulatory protein that is acting as a binding for the specific DNA sequence. Okay, southern western blotting as the correct answer. Sorry, southwestern blotting. Okay, and the next question, which of the following disease in human is caused due to expanded microsatellite repeats? Which there are four options: Huntington disease, cystic fibrosis, tuberculosis, Alkaptosuria. Okay, so these are the four different options they have given. We need to identify the microsatellite repeats. Okay, so the option A will be the correct answer. Okay, apart from that, there are different kind of disease like myotonic dystrophy and amyotrophic lateral sclerosis and the Huntington disease, HD disease. So these are the example of the microsatellite repeats. Okay, so the option A will be the correct answer. And the next one is, which of the following disease is associated with Philadelphia chromosome? Okay, so there are retinoblastoma, arterial septal defect, chronic myelogenous leukemia, myotonic muscular dystrophy. So from this, we need to identify which disease is associated with this particular Philadelphia chromosome. Okay, so the option C will be the correct answer. Okay, so this particular disease will be found in the bone marrow cells. Okay, so the people who have the chronic myelogenous leukemia will be affecting with the Philadelphia chromosome. Okay, and almost and some people will have the acute lymphocytic leukemia and acute myelogenous leukemia. Okay, so all these conditions are due to that Philadelphia chromosome. Okay, so the option A will be the, sorry, option C will be the correct answer. And the next one, the classification system that groups organisms together based on the similarity of their observable characteristics is known as. The option A, phylogenetic system, phenetic system, phyletic system, and complement system. So the option A, phylogenetic system, it will show the similarity of evolutionary relationship. Okay, and the option C, it will also show the evolutionary connections and evolutionary changes. Okay, and the last option, complement system, it is completely based, it will be on the immunity side. Okay, this is an innate immunity which is responding against the antigen. The remaining option is what? Option B, phenetic system, which will be helpful for the identifying the similarity of observable characteristics. Okay, for the appearances, this phenetic system will be helpful. So, okay, so the option B will be the correct answer.